Assalamualaikum warahmatullahi wabarakatuh. How are you doing? Hope you guys in a good state. We are from group 4 and our group member consists of Alia, Ivana, Akma and Farah. So in this video, we will be presenting a topic based on article entitled Epigenetics in Ovarian Cancer. Hope you enjoy this video. Let's start. For introduction, there will be two terms that will be explained thoroughly. Firstly is ovarian cancer and secondly is epigenetics. Ovarian cancer is a disease that have poor prognosis and have diagnosis at a late stage and have high rate of resistance to the standard chemotherapy. There are four types of epithelial ovarian cancer, which is HGSOC, ENDOC, CCOC, MOC, and LGSOC. Let's move on to epigenetics. Epigenetics is the dynamic regulation of gene expression by DNA methylation and histone post-translational modification in response to environmental cues. For the summary of epigenetics, epigenetic regulatory elements are comprised of post-translationally modified cytosines, non-coding RNAs, and histone modification. The histone modification is by through HDAC, which is acetylation of histone, and it is overly expressed in ovarian cancer. It is heritable and regulate the means by which the genome and environment interact. The most common study is DNA methylation and histone acetylation. So in this article, they are more focusing on DNA methylation. DNA methylation is playing a role in imprinting cell differentiation during development and resulting phenotypic variability. There are two types of methylation, which is hypomethylation and hypermethylation. In ovarian cancer, hypomethylation incidence can cause aberrant expression of repeat elements and oncogenes. Meanwhile, for hypermethylation incidence, they can cause silence genes that regulate critical functions. For DNA methylation in epithelial ovarian cell, or EOC, study focus on the methylation state of specific sets of genes and genome-wide DNA methylation, comparing EOC histotypes and normal ovarian tissue. For targeted analysis, it was found that formerly DNA methylation analysis in EOC were targeted to genes commonly mutated in cancer. It was also found that most commonly studied are the high-grade serous ovarian cancer. In most analyses, they use methylation-specific PCR to evaluate the presence of PCR fragment amplified with primers that target the preserved methylated cytosine in bisulfite modified DNA. For genome-wide analysis, the Cancer Genome Atlas studies use normal ovarian or fallopian tissue as the control, while other studies also include hierarchical clustering to discern the methylation pattern within EOC cases. The most commonly studied ovarian cancer are high-grade serous ovarian cancer, endometrial ovarian cancer, and clear cell ovarian cancer. In high-grade serous ovarian cancer, or HGSOC, TCGA had identified 168 genes with altered gene expression in HGSOC compared to normal fallopian tube, with 11.5% hypermethylated at BRCA1 gene. In another analysis, it was found that 543 hypermethylated genes with decreased gene expression, with 5 of those genes overlap with those identified in the TCGA dataset. In endometrial ovarian cancer, or ENDOC, soft CpG loci were differentially methylated in both HGSOC and ENDOC compared to normal tissue. All but one CpG had reduced methylation in tumor. Hypermethylation also was noted in a CpG island downstream of the transcription stat site for ZNF154. However, not all ENDOC in this analysis showed hypermethylation at ZNF154, with some ENDOC cases having a low-intensity methylated phenotype. Overall, ENDOC appears to have a significantly different methylation profile compared to HGSOC, with most having a CpG island methylated phenotype. For clear cell ovarian cancer, or CCOC, the study identified specific dysregulation of genes that are involved in oxidative stress response, including HNF1B and VCANN. While HNF1B is overexpressed in CCOC, however, it is methylated in 50% of HGSOC. In summary, both CCOC and ENDOC are most likely to have a hypermethylated phenotype, while HSGOC is hypomethylated and has fewer hypermethylation events. CCOC and ENDOC also are thought to arise from endometrial epithelial and endometriosis, suggesting that inflammation may play a role.
The next part is on chromatin alterations in ovarian cancer. Studies has identified mutations in ARID1A, SPOP, and KMT2D genes. These genes involved in chromatin remodeling, so the mutations of these genes in clear cell carcinoma suggested that chromatin has been remodeled in these histotypes. Next, in epithelial ovarian cancer, histone post-translational modifications and histone PS kinase has been evaluated using immunohistochemistry and next-generation sequencing. The result shows upregulation of SIRT1 gene in invasive serous epithelial ovarian cancer, and also reduction of H3K27MA3 expression. It is also found that the reduction of H3K27MA3 expression is associated with decreasing rate of ovarian cancer. Lastly, H3K27MA3 and H3K4MA3, which are active and repressive histone markers respectively, has been studied in high-grade serous ovarian cancer. The results include lower expression of bivalent and H3K27MA3 marked genes. And out of 580 bivalents, 355 of them are found to be tumor-specific. Plus, the methylation of H3K27 protein is mediated by a polycomb called PRC2, where PRC2 complex gene will support the methylation process and lead to gene silencing in high-grade serous ovarian cancer. As a summary, tumor progression and tumor resistance are influenced by saturation of chromatin in some cases of epithelial ovarian cancer, and also the presence of tumor-specific bivalent histone marks.
Next are the summarized for clinical aspects of epigenetic in EOC. Firstly, the chemoresistant. One of the resistance is due to DNA methylation because it is one of the mechanisms cells to respond to the environmental stimuli. For this, the platinum-based chemotherapy is used as the primary line of drug therapy. This is because cancer cell lines were reported its sensitivity to platinum-based chemotherapy in cells. There is a research found out there are 19 genes associated with carbaplastin resistance, where it reduced the carbaplastin sensitivity as one of the chemotherapy medication. The study revealed overlap in the genes identified for chemoresistant or survival, as development of chemoresistance is a complex process and involves more than a single gene. Second are epigenetic inhibitors in EOC. DNA demethylation agents such as FAF-AZA cysteine AZA was used to express gene silence by DNA methylation. This AZA enhanced the sensitivity of the platinum-resistant ovarian cancer cell lines. The HDAC have been evaluated in cell lines where it promotes the NMTI protein degradation and inhibit the DNA methylation. Also found out that decitabine treatment for anti-tumor are varied up to no effect for ovarian cancers. For BET inhibitor, it inhibits the binding of the histones, acetylated lysine, thus preventing gene transcriptions. However, resistance in BET inhibitors also have been reported. HDACs have been reported to sensitize platinum-resistant EOC cell lines, thus it undergoes some clinical trials to develop more selective HDACs that targeting histotypes. Next are about cell-free DNA. They use liquid biopsies to detect cell-free tumor DNA in most advanced malignancy. It can also detect aberrant levels of methylation in genes, and also for screening high-risk patients for the presence of cancer. They are undergoing clinical trial using carboplatin or toxoid in EOC where they found out it evaluate MLH1 methylation in EOC plasma, which is associated with poor overall survival. Fourth are about molecular methylation profiling in EOC, where it is to understand and align differences in survival time between different genes. They identified 4 methylation profiles significant such as C1 and C3 where they show higher global methylation and longer survival. Secondly is to know the response to the therapy by seeing in the histological subtyping of the ovarian cancer. Thus, the molecular methylation profiling is important as it can be used to build predictive models to communicate personalized response to the therapy.
We have come to the last section in this video, which is conclusion. As for conclusion, the aberrant histone acetylation and DNA methylation can alter the gene expression, lead to consequence of malignant incidents. However, the epigenetic changes are reversible and easy to modulate. The epigenetics may serve as basis of development of diagnostic tools aiding in early detection of ovarian tumor. Therefore, for future research, the epigenetics-based biomarker profiling and therapeutic regimens may provide a powerful weapon to fight against ovarian cancer. That's all from us today. Thank you. Assalamualaikum.
All day, all day, all day.